In this work, we introduce a dataset named ProBio, the first protocol-guided dataset with dense hierarchical annotations in molecular biology lab. We further propose two benchmarks: transparent solution tracking and multimodal action recognition. Is there a reproducibility crisis in science? Nature has asked 1,500 researchers this question as part of an online survey. Half agreed there's a significant crisis and many more identified a slight crisis. Science moves forward through corroboration. But 70% of researchers who took our survey said they tried and failed to reproduce another group's experiment. And even when trying to reproduce their own work, just over half said they couldn't. Does this mean that most published research is wrong? Our respondents don't think so. Most still trust published results in their field, with chemists and biologists the most confident. There are many reasons why researchers may fail to reproduce results. Most times, a small difference like shaking or stirring a sample can lead to different outcomes. According to the survey of Nature and Science, only 36% of studies can be replicated, and the main factors are listed below. It is worth noting that the irreproducibility caused by deficiencies in methods and experimental design is widely considered to be very important. In the biology domain, the AI assistant is equal to the protocol-guided monitoring system. Toward building such an AI monitoring system, multimodal dataset in the specialized domain is needed. The task of delineating intricate action hierarchies for daily activities is challenging. Some work justifies action hierarchy design by examining activities in sports and kitchens. The annotations provided, while detailed, may lack strong contextual information and may not be suitable for complex tasks that demand nuanced multimodal understanding. Nonetheless, the practical applications of these tasks are limited. Our ProBio dataset provides aligned video protocol pairs, accompanied by detailed experimental instructions for every procedure. Benchmarks on ProBio offer a comprehensive examination of existing models for multimodal video comprehension in specific domains. The left image shows a panoramic view of the laboratory taken in our cooperative international standard laboratory, along with camera settings. The right image is a schematic diagram of camera settings in another standard room. The video on the left showcases our self-built website, aimed at enhancing the annotation efficiency and interactivity of professional researchers worldwide. The goal is to expand the brief experiments downloaded and pre-processed from top-tier platforms into executable practical experiments. The video on the left is a labeling tool we designed ourselves, with the purpose of establishing a correspondence between the current state of videos and practical experiments by assigning action labels. The video on the right is the result of acquiring human-object interaction pairs under the guidance of practical experiment alignment. Finally, our ProBio dataset contains the following content, as shown in the table. There are two subsets for different benchmarks, 3 levels of ambiguity, and a 3-level hierarchical structure. The specific distribution and hierarchical relationships are shown in the figure on the right. The first task focuses on transparent solution tracking. The following 4 images display some transparent objects and solutions in the biological laboratory. The input and output of our task are listed below. The following table shows the tracking results for solutions in both vision-only and protocol-guided scenarios. We observed that visual tracking in TransST is challenging. More robust object detectors benefit tracking in TransST, and understanding protocols is crucial in TransST. The 3 videos below display the tracking results in the form of masks overlaid on the videos. The second task focuses on multimodal action recognition. Actions in the biology lab exhibit strong ambiguity. Specifically, similar movements can represent entirely different tasks and lead to divergent purposes. As shown in the following 4 sets of videos, seemingly identical actions have different labels. We use the bidirectional Levenshtein distance ratio to define ambiguity and calculate the ambiguity values for all practical experiments, as shown in the right figure. The table below shows the recognition of actions in 5 different scenarios. We found that actions are visually ambiguous in multi-AR. Recognition in multi-AR demands a detailed understanding of protocols. Contextual information is crucial for multimodal understanding in multi-AR, and solving protocol-level ambiguities is a bottleneck for multimodal video understanding in multi-AR. Below are the success and failure cases of action recognition using our method. Overall, we introduce ProBio, the first protocol-guided dataset with dense hierarchical annotations in the biology lab to facilitate the standardization of protocols and the development of intelligent monitoring systems for reducing the reproducibility crisis. We propose 2 challenging benchmarking tasks, and we provide an extensive experimental analysis of the proposed tasks to highlight the limitations of existing multimodal video understanding models and point out future research directions.